First, let me introduce myself. I'm Friederike Ehrhart. I'm a postdoc researcher at Maastricht University. I have a double affiliation both in the Department of Bioinformatics of Chris Evelo and in the Dutch Rett Expertise Center with Leopold Curves. What I would like to present you today is an ELIXIR-funded implementation study about molecular data in rare diseases. And this is a collaboration of ELIXIR Spain and ELIXIR Netherlands. The collaborative partners are Maastricht University, of course, Leiden University Medical Center, and Barcelona Supercomputing Center. And the main PIs involved here are myself, Chris Evelo, Marco Roos, Annika Jacobsen, and Salvador Capella. Our implementation study took, just for basic view to know, it took 12 p.m. and started last year in April and ended in December last year. So I'm presenting you here the outcome of our tasks. One of our tasks was first to get the status quo of rare disease and molecular data to see what is actually here. Second task is to assess their status of interoperability and the workflows for verification. Then we had a look at what can we actually do to harmonize data across different sources to develop a workflow and see what we can do with the data, to create and improve resources, what we need and what we can do for updating this. And task 5 is actually an application use case. So what actually can we do with the gained data here? What can we do to solve scientific questions related to rare diseases? So speaking about rare diseases, the use case I'm talking about will be Rett Syndrome. Rett is a rare monogenic disorder which is caused by a loss of function mutation from the methyl-CpG binding protein number 2, MECP2. The diagnosis criteria are usually stagnation and the loss of acquired motoric and communication skills at the age of about 6 to 18 months. And the phenotype of the adult or the kids with Rett syndrome is that they have a severe mental and physical disability. They usually show stereotypic movements, so they usually do this hand-wringing stereotypic movements. They often have epilepsy, an abnormal muscle tonus, and develop during their lifetime often scoliosis. So when we speak about MECP2, that gene is actually very important for the development of the neurological system. And since a few years ago, it was discovered in '99, only very few things were known about this. When in 2016, we did not know anything about the interactions about MECP2. I started summarizing them and making a pathway about that. It's actually rare diseases are not only interesting for people who actually suffer from the rare disease, but also for which teach us about the normal gene function in the normal human physiology. So it's actually interesting for all of us to study rare diseases. So when we talk about molecular data for rare diseases, we are speaking about very different sorts and very different classes of data. One of them would be patient data, which is usually collected in patient registries. These usually, as they have sensitive data, they are usually with limited access. They are often offline collections at clinics, in hospitals, or at individual researchers or even individual doctors. Their interoperability status is therefore often not very good. So you usually are confronted here with Excel sheets or Word documents or hospital internal documentation files. So there's no standard format, and they are very different from the input and the form. Then you have here the omics data repositories. And these are things like EGA is for, where you put really large-scale raw and processed omics data for studies. Then there are the genotype-phenotype databases. These are often collections of genotypes which are associated with a certain phenotype. That can be a diagnosis, that can be a collection of phenotypic terms like phenotypic ontologies, or just descriptions, or just a SIFT or a FEN score. And then, of course, we have databases for individual proteins, genes, etc., with or without mutations. So this is actually the broad variety of data what we have when we say we are dealing with molecular data in rare diseases. So when we started the implementation study, these were our starting points here. I sort of inherited it from a doctor who is about to retire, quite messy spreadsheet, an Excel sheet with patient data, genetic information, diagnosis, care score. The care score is a score to assess the physiology and the individual abilities of Rett patients. So it's of course adapted not to collect the full phenotype of a patient, but the phenotypes which are typical for that disorder. And then personal notes from the doctor who created that spreadsheet. So here our task was to verify this data and ideally also to share it with other researchers in RD Connect or via OrphaNet. Then speaking about genotype-phenotype data, so with not only the detailed phenotypic description, see what is there with the data, see how the upload and download functionality of the different genotype and phenotype databases is, get the existing information and try to integrate the data among the different databases and see what additional information can we get if we use not only one but all or a lot of different sources. Then we try to do a resource update and improvement. We used wiki pathways for molecular pathways for rare diseases. We had a look at gene variant mapping databases. We added an OMIM gene disease link set, and we're thinking about adding a Campbell drug target link set for the development or drug development within rare diseases. Then that's what the Maastricht Rett Syndrome data set looked before I started with it. We had about 324 patients in the registry. I must say this is a historic data set, so we did not start to collect it. This was a collection of a doctor who did that the past 20 years. And it contains genetic data, diagnosis and phenotypic data, and individual annotations. Several patients visited the hospital multiple times, so we have more than one entry for one patient. Yeah, my goal here was to make this data set interoperable or fair. The biggest problem here at the end that I learned was that I'm not allowed to share this patient data, even not in access control databases like RD Connect, because they are not recognized by the Dutch legislation as safe resources or safe resources where you can put sensitive patient data there. So in Netherlands, they are very restricted. I can only do that with an ethical approval and with informed consent of the patients or respectively the caregivers. So for historic data, this is at the moment not possible, but this is something we definitively keep in mind when applying for new studies, so that the new data which is collected from now on can be stored in RD Connect. And the verification process of the dataset itself, that took quite some steps. The genetic information there was in quite different standards, so I had to bring them all in one standardized HGNC format in one reference sequence. The diagnosis information was annotated with OMIM terms. Phenotype information and care score were mapped to the human phenotype ontology, as well as the individual annotations and observations. So as far as possible, I annotated them with the proper HPO terms, and if there was medication information, I added the drug bank ID for this. So the biggest problems I experienced here was that the data capture spreadsheets from the clinicians, they come with very different and very individual input information. So they're using a broad variety of formats, lab slang, historic formats, different reference sequences, which I had to standardize. And so I also often ask experts which observed the genetic information annotation for not the past 10 years, but the past 30 years. Then also the task was to translate the medical technical language, the slang as I would say so. Even as a biomedical scientist, I could not always differentiate between the terms they were using there and terms I find in Wikipedia or any other medical information sheets. So I had to sit down with the experts and ask them, what do you actually mean by this term, and can we find a proper term in the HPO ontology? So now we have our interoperable data set annotated with genetic and phenotypic information in all the proper formats. So what can we actually do with that? This is one possibility. I mapped here the genetic information of one patient with a 925C to T mutation. And here in the center, the yellow dot is the genetic information, and the white dots are the phenotypes described for this one patient. All, of course, annotated with the proper ontology. Then, if I go one step further, I have here more patients with the same mutation, And now I can actually start counting how often is one phenotype described within that patient population with exactly this mutation. You see the red dots, the more redder it is, the more often it is observed in patients. If it's bright red, then it is more than 50 patients. So this is a quite abundant mutation here. And if I go even further and have a look at all of the mutations I have, here I integrated not only the Maastricht-Rett dataset, I downloaded the available information from the ELIXIR database. They had also 35 patients with mutations in MECP2 and added their annotation too. So what we see here in the center are the 79 different mutations and information about 356 patients. And what we actually can see here at the bottom, where all the red dots are, the phenotypes which are described most abundantly, they actually match quite close to the diagnosis criteria of Rett syndrome. So this actually can become very interesting for look for overlap between diseases, between different disorders, and also to see what is actually the most abundant phenotypes, what are medium abundant phenotypes, and what are rare phenotypes of this disorder. I must say at this point that Rett syndrome is maybe not the best example for application of this kind of analysis. MECP2 is on the X chromosome, and the X deactivation or inactivation degree plays also a big role in the disease severity and the severity of the disease phenotype. But for any other disorder or genetic disorder, this can become, I think, quite interesting. So to now, I was talking mostly about genotype and phenotype information, so with detailed phenotype descriptions like the HBO terms. Genotype-phenotype databases contain actually only one I found which contained that level of exactness. The other genotype-phenotype databases I looked for, so findability is not that high. Only up to about 2/3 of these list of databases I found via fair sharing. The other ones via personal recommendation or studying the Rett-specific literature. From all these, we have quite a lot of different databases here, and they become even more different when you look at what they provide as actually phenotype information. RettBase, KMD, ClinVar, and Café Variome provide usually a diagnosis, like Rett syndrome. The ELIXIR is the only one which provides a detailed phenotype, and LOVD, EVS, EVA, EXAC, DBSNP, DBVAR, and even some more, they give only a score. So they give the genetic variation and a predicted score about the potential pathogenicity. The problems if I want to integrate genotype-phenotype information about all these different sources and try to figure out what we can actually do here, that was quite interesting. First of all, we had a lot of broad variety of different genetic formats. Most of them were actually using HGVS or RS numbers, but all of them had different reference sequences, or were using either the mRNA, the protein, or genetic position. And I also had to deal with some of the historic formats. For this, we tested during the implementation study several liftover tools which can translate between the different formats, and found that actually the best was Mutalyzer. So you can use a batch format or a batch, say upload a spreadsheet, or type in individual mutations and ask for translation into a different format, a different reference sequence. The next problem I experienced here was the consequent application of these formats. When looking at the different databases, I have here the databases which have disease, that have a diagnosis annotation. Then we see here some Maastricht-Rett dataset, the ClinVar, RettBase, KMD, and EVS partly. And they have, this is the number of MECP2 variations they provide. The criteria, diagnosis criteria, which I choose for, this is Rett syndrome. These are named here. And we have here the number of variations meeting that criteria, so actually Rett-causing mutations. And after using Mutalyzer, I was able to translate them in a different format for between 89 and 100%. The rest of all of them had some nomenclature issues or not enough information that I can actually get the exact information. After manual curation, I was able to increase the number to something like 91 until almost 100% here. I must say here that the Maastricht Rett data set is historic data set. For some of the patients, we don't have the exact mutation, or only an annotation like this is a gross chromosome rearrangement, or here exon 1 and 2 is missing. So this is not an exact information I can put in here. I must also say that all the other databases have been seen by a bioinformatician or by an expert before, and with the Maastricht Rett dataset, I was maybe the first one who was actually looking into deep here. Yeah, so with the consequent application of the format, that's actually also something where teaching and bring your own data sessions can help to improve the knowledge of curators to apply these formats consequently and make it better and easier accessible for others. Then the problem was also the low degree of interoperability of the phenotypic data. I found the use of ontologies or ontology terms or identifiers actually only 2 times in the KMD, the Korean mutation database. They used DOID, the disease ontology annotations for Rett syndrome, and the CIFR. And the others, well, disease information or pathogenicity scores. Then another problem was also, well, the reuse or data accession permission was often hidden somewhere in the documentation and also termed a bit vague. So there was no CC-BY or anything copyright information. Some of them stated just that this is should be used for clinical research and is not for patient information, but actually no real terms about can we reuse the data, can we actually look at the data. So finally, integrating all the data from the different sources with annotation of this is a Rett syndrome causing variant. We have now a data set with about 4,000 variants, 828 unique variants. Many of the hotspot mutations are actually there multiple times, so they make up of about 60% of all Rett cases. And we have a second spreadsheet with 400 benign MECP2 variations. And they are currently available as a spreadsheet, but I hope we have that as an RDF file soon. Then, using these spreadsheets, we tried to analysis and model the data and see what we can get out of that. From the 4,000 Rett-causing variants, we found that, well, quite as we expected, that we have a lot more, using the variant effect predictor tool from Ensemble, we have a lot more missense and stop gained and frame shift variants than in the benign variants. So this is not that much surprising. What was actually more interesting is that we found if we have a look at the predicted pathogenicity scores using meta-LR for a score, we have here at close to 1, this is pathogenic, and close to 0 is expected to be benign. That we have here in the Rett-causing population, some which are predicted to be benign, but still were annotated with Rett syndrome or found in a patient with Rett syndrome. We have in the benign population some which were actually predicted to be very pathogenic and still do not cause a disease. And to our big surprise, we found even overlapping variations. So variations which were found in both spreadsheets, the pathogenic and the Rett-causing and the non-Rett-causing population. Yeah, we are still analyzing the data, and I hope we can make a publication out of that in the following year. What possible explanation would be here, we don't know anything about the patients behind the data. This with a benign, in the Rett-causing populations with a very benign meta-LR score, that could be, for example, male Rett patients. And the pathogenic ones in the neutral population, they could be female carriers, so we don't know for sure. So then, almost finally, we worked also on resource update and improvement of data for rare diseases and tools for rare diseases. During the implementation study, we created in Wikipathways 3 new pathways for rare diseases. We include now Prader-Willi syndrome and Angelman syndrome, rare prion disorders, and HDAC6 interactions. HDAC6 is a molecule which is associated to Rett syndrome and other neurological disorders. We figured out a way how to add genetic variation data into a pathway. One possibility would be adding the information in form of the GPML file, currently without visualization. And we created, also for a previous implementation study, a BridgeDB dataset, which links genes to variations. This is available in the BridgeDB homepage by now. Then we were also working on link sets, spreadsheets, and data sets. So we created the gene, disease, and provenance link set for OMIM information. So this gene causes that disease. Then we were working on a Cytoscape add-on. Cytoscape is an important tool for network analysis. But there is an add-on, it's called CyTargetLinker. I can show you quickly here what it does. It extends actually existing networks with information from external databases. So you can ask, for example, another database. This is if you look at this network, do you recognize any genes which are targets of transcription factors? And if yes, give me the transcription factors and visualize them. And we did that for Campbell. So we have now a data set which can actually recognize drug targets or chemical compound targets, which is actually interesting for rare disease drug development, because maybe not all drugs are yet registered in drug bank or are experimental drugs which are not used here. So we have here now a broader variety of chemical compounds which can act on molecular pathways. So finally, I would like to give you a short summary. So what did we actually do in that implementation study? We investigated the current status and problems of the molecular data for rare diseases. We improved the interoperability aspects of these diseases, so by variant to phenotype mapping and visualization. We have now a better ontology-based annotations of data for rare diseases, especially our data set, but this can also serve as a blueprint for other spreadsheets and other information for other diseases. And hereby we enabled the modeling of larger amounts of genotype-phenotype relationships. The problems we identified during this implementation study is that we often suffer from different minimum information standards or very different levels of information which are recorded by the experts and put into databases. Many of the data is not yet mapped to ontologies, or phenotypic data used for diagnosis is often inconsistent, and description of the diagnosis itself is also not always agreed among the experts. Here, historic data can often be improved manually if enough minimum information is available, but we actually recommend that new data records should be assisted, so that we provide clinicians with pull-down menus or capture frameworks, or, for instance, also write fields can be used for that. Another problem was also the broad variety of formats in which genetic information is stored in these databases, current and historic formats, but we actually also found that Mutalizer can often fix that. Consequent application of the nomenclature is an issue. Something between 5 and 12% in the database entries are not correct, or not correct nomenclature, and this actually could be improved by teaching events. And the reuse of the data accession permissions often hidden, and that should also be put more into the front and clearly state which data can you use to what conditions. Then, some gold nuggets we discovered during the implementation study. The database LOVD actually was the only one which provided different genetic information formats at once, and had Mutalizer as a part of its toolset included, so that you immediately can translate between the different genetic formats. Very handy when it comes to translation or integration of data across different sources. BYODs helps to encourage the correct application of the standards, showing their practical usefulness and actually bringing clinicians and researchers immediately to correct input of the database. Then we created a few silver nuggets, I call them. The BridgeDB gene to variant database, which is already available. The CyTargetLinker, the Campbell add-on. If you move pathways for wiki pathways, and data link sets and spreadsheets. They are available in the last page. I put a list of all of these. So at the end, I would like to thank you for your attention. Thanks to my collaboration partners of the implementation study, especially to Annika, Marco and Raja, Salvador, Maria and Mattia, and within Maastricht University to Chris, Johnson and Ryan. And also special thanks to the head of the Maastricht Rett Expertise Center, Leopold Kurfs, then Henk van Kranen, Jeroen Laros, David van Enkevoort, and Mark Wilkinson. Thank you.